Greetings to all. My name is Dr. David Jett, and I'm the Director of the Countermeasures Against Chemical Threats program at the National Institutes of Health. This is also known as the NIH CounterACT program. I'm here along with Dr. Chardell Spriggs, who is the CounterACT program analyst. Today, Chardell and I will present a short webinar that describes several funding opportunity announcements recently released by the NIH CounterACT program. The webinar will provide a short introduction that describes the program, followed by a description of 4 announcements that are published on our website. The focus will be on the goals and scope of the different announcements and special considerations when writing a grant application. It is hoped that this webinar will help potential applicants decide which funding announcement is best for them and lead to the development of more competitive applications. Mission of the NIH CounterACT program is to understand fundamental mechanisms of toxicity caused by chemical threat agents, and the application of this knowledge to develop promising therapeutics for reducing mortality and morbidity caused by these agents. What are chemical threats? We have 3 categories of higher priority chemical threats. The first category is traditional chemical warfare agents, such as the organophosphorus nerve gases like sarin and VX. The second are the toxic industrial chemicals such as chlorine and cyanide. And the third category are the toxic agricultural chemicals such as the insecticides aldicarb and chlorpyrifos, and rodenticides like sodium fluoroacetate. There are 27 institutes and centers at NIH. The CounterACT program is a partnership among several of these that have expertise needed to cover ocular, skin, pulmonary, and other effects of chemicals we study. The program has many moving parts that make up a comprehensive, coordinated effort to advance the mission of supporting research on chemical medical countermeasures. The grant program engages many academic laboratories as well as government and commercial labs working in this area. We have an extensive collaboration with the U.S. Army Medical Research Institute of Chemical Defense, which is the premier military agency in this field. This institute, as well as the CounterACT Efficacy Research Facility, are critical to our mission because they can do studies with chemical warfare agents that can only be used in approved facilities. We also have the CounterACT Preclinical Development Facility that conducts safety and pharmacology studies. These contract facilities support the NIH and NIH-funded investigators in product development. Our current funding announcements include exploratory small grants to conduct basic research on the toxicology of chemical threat agents, to advance our scientific knowledge that is so critical to understanding therapeutic approaches, and other studies to obtain preliminary data for application submissions to more advanced funding announcements that include a lead identification funding announcement, a lead optimization funding announcement, and a research centers of excellence solicitation. These will be discussed in the following slides. There are 2 important things I wanted to mention here. One is be sure you are following the most current funding opportunity announcement, and these announcements can be found at our website listed here.
The first FOA that I will briefly describe is the CounterACT R21 program. In general, the R21 grant mechanism is intended to encourage exploratory and developmental research by providing support for the early and conceptual stages of project development. The proposed studies should be novel and break new ground in the field. These studies can also focus on extending previous discoveries toward new directions or applications. As such, the main goal of proposals responding to the CounterACT R21 FOA is to generate tools and preliminary proof of principle efficacy data to support the identification of potential models and/or therapeutic hits that could lead to the development of small molecule or biologically compounds. Typical scope of CounterACT R21 studies includes creation and validation of screening assays, identification of candidate therapeutic targets and biological markers, and demonstration proof of principle efficacy. Applications in response to the CounterACT R21 solicitation are only accepted once a year. The submission deadline is at the end of January. Proposals are reviewed the following July with an anticipated project start date in September. It is important to note that R21 projects can only be supported for 2 years and cannot be renewed. The combined budget for direct costs for the 2-year project period may not exceed $275,000. No more than $200,000 may be requested in any single year. There is no limit regarding how many active R21 projects a PD or PI may have at any time, but all projects should be distinct from those that could be supported through other more traditional mechanisms, such as an R01. The next couple of FOAs that we will describe are those supported under the Cooperative Agreement Research Mechanism. Again, cooperative agreement projects are milestone-driven grants that will involve substantial programmatic involvement in the research activities between CounterACT staff and the awardee. The first cooperative agreement FOA is the identification of therapeutic lead compounds. As the title implies, the main goal is to identify small molecule or biologic lead compounds that are excellent candidates for therapeutic development. Research that can be supported under this FOA includes confirmatory studies of molecular targets that would inform on specificity, affinity, potency, and selectivity, demonstration of both in vitro and in vivo efficacy, as well as preliminary preclinical studies such as safety, toxicity, pharmacokinetics, and pharmacodynamic studies. Applications in response to the CounterACT Identification U01 are only accepted once a year. The submission deadline is in the middle of September. Proposals are reviewed the following March with an anticipated project start date in the following July. The proposed budget for direct costs for the identification FOA is capped at $300,000 per year. The proposed project period is limited to 3 years, and renewal of the project is not accepted. There is no limit on how many applications an institute may submit in response to the identification FOA. However, the submitting PD or PI of the applicant organization is limited to no more than 1 active individual or multiple PD, PI identification award at a time. The second CounterACT Cooperative Agreement FOA is the optimization of therapeutic lead compounds. A previously identified lead compound where specificity, affinity, potency, target selectivity, efficacy, and safety have been established is a prerequisite for this program. The main goal here is to optimize the pre-identified lead compounds so they are ready for advanced development activities that lead to regulatory approval and licensure. The scope of research that can be supported under this FOA should aim to generate in vivo efficacy data in human-relevant animal models consistent with the intended use of the product in humans, including the likely environment where the drug would be administered. In addition to efficacy, the optimization U01 could also include bioanalytical assay development and validation, demonstration of laboratory scale and scale-up manufacturing of the product, and non-GLP toxicity and pharmacology studies. Similar to the Identification U01 program, the Optimization U01 also only accepts applications once a year. The submission deadline is the same in the middle of September. Proposals will be reviewed the following March, but separately from the identification responses, with an anticipated project start date in July. Unlike the identification FOA, the expected budget for direct costs is expected to be between $300,000 and $500,000 per year. Project periods may be proposed for up to 5 years, but renewal of the project will not be accepted. Again, there is no limit on how many applications an institute may submit in response to the optimization FOA. However, the submitting PD and PI of the applicant organization is limited to no more than one active individual or multiple PD, PI optimization award at a time. As a reminder, the main goal of the Optimization UO1 program is to produce a single optimized lead compound that is ready for advanced development activities that would eventually lead to regulatory approval and licensure. These subsequent advanced development activities could be supported by another agency within the federal government as well as by the pharmaceutical industry. While not a guarantee, in order to secure the interest of an advanced development, an optimization UO1 project should culminate with one lead compound where the ADME, the absorption, distribution, metabolism, and excretion properties are well understood. Just as critical, efficacy must be demonstrated with both the experimental animal model and actual concept of use the therapy is directly relevant to in humans. The compound can be stably synthesized and scalable. It will also be important that a sound preliminary regulatory strategy be in place with the appropriate experts identified. Finally, the project must have secured the relevant intellectual property rights on the compound by the end of the project period.
So, in addition to the UO1 programs, we also have a centers program, Research Centers of Excellence. These are U54s. And the scope of this program is similar to the UO1 programs as well. It's inclusive, a bit more inclusive. It includes target and candidate identification and characterization through optimization and demonstration of in vivo efficacy consistent with the product's intended use in humans. These are large multi-project programs. The center structure includes one administrative core for center management and operations, 3 to 5 interrelated research and development projects, up to 3 scientific cores to support these projects, and 1 research education core. The Research Centers of Excellence are overseen by a steering committee as well as an external advisory committee. The application due dates are in September, mid-September. The budget may not exceed $2.5 million in direct costs per year, and the project period may not exceed 5 years. For these research centers, the project should culminate with the same criteria that you heard about in the optimization UO1 program: 1 lead compound with well understood ADME, efficacy in an appropriate animal model, stability and synthesis information, and a regulatory strategy and intellectual property. So, we're going to end the webinar with a few additional considerations when preparing an application. There are 2 NIH-wide specific things that you must address now with all NIH applications. One is enhancing reproducibility through rigor and transparency, and there is a notice that you should look at before you write your application, and this includes things like adequate scientific rigor, statistical analysis, control of bias, and so forth. So please be sure to read that notice. And the other one is consideration of sex as a biological variable in NIH-funded research. Again, you need to address effects of sex and age alone or in combination as biological variables. A few CounterACT specific considerations include practicality in real-world civilian mass casualty situations. The product should have utility for emergencies involving acute chemical exposures where medical intervention is required immediately in the field or in hospital. Secondly, special consideration will be given to research relevant to subpopulations with higher vulnerability, such as pregnant women, infants, and so forth. And then lastly, we always encourage working with drugs that are already approved by the FDA for other indications for repurposing those drugs for chemical threat indications. Fourth, research strategy. It's important if you're working with novel chemical structures to include those chemical structures in your application so they can be reviewed through the peer review. Secondly, regulatory strategies addressing central elements relevant to FDA guidelines, and those FDA guidelines include things like the animal rule and so forth. So it's important to read those guidelines before you write your application. Milestones. These are milestone-driven projects, all of the U01s and as well as the U54s, and these milestones should be quantitative. They should be a description of what constitutes successful research progress and development. They should describe the goal of the work and not just a statement that the work will be completed. And we urge you to look at our website for an extensive discussion on how to write appropriate milestones. And then finally, work closely with your institutional tech transfer people to discuss intellectual property and patents. So here's a list of the CounterACT program staff at NINDS in central office, as well as some of our partners across the NIH. And of course, always go to our CounterACT website, www.ninds.gov/CounterACT. So we thank you for listening to us, and please feel free to contact us if you have any further questions.